Next speaker is Ian MACLACHLAN, who is going to talk about adaptive genomic variation in lodgepole pine. Ian?
Thank you. Hey, good morning. Thank you for inviting me over to share some of my experiences from research in Canada, despite the sound of my voice. I'm going to talk about some of the results that came out of the latter stages of my PhD with Sally Aitken. I finished this a couple of years ago, and I've been sort of refining and improving the techniques and the results from this for the last couple of years. Looking at genomic adaptation in lodgepole pine and what the implications of selective breeding are on that adaptation. This is a map of British Columbia and Alberta in Western Canada. You've got historical climates on the map to the top. We've got the projected mean annual temperature from the 2050s in the map below. You can see that warming is predicted even under a moderate climate change scenario. To combat the effects of this in terms of forest management, assisted migration policies are being implemented in Western Canada. These assisted migration policies use the existing breeding zones, and they move reproductive material, seedlings, around to match future climates. However, layered on top of this assisted migration policy is selective breeding. Selective breeding aims to shift the distribution of favorable traits, in this case, height growth for timber production, but associated with that, we expect correlated responses to selection in other adaptive traits. And so the concern here is that by implementing selective breeding and favoring height, we generate negative correlated responses to selection that then compromise our ability to implement assisted gene flow and mitigate the effects of climate change. So the primary research questions: one, what are the genome-wide effects of artificial selection for increased timber production, i.e. height growth, on climate-related traits in lodgepole pine? What are the genetic architectures of polygenic adaptive traits in lodgepole pine? And are there implications for assisted gene flow from genomic adaptation and genomic responses to selection for height growth? Sampling. We sampled 12 breeding zones from across British Columbia and Alberta. These breeding zones represent second to third generation breeding programs. We sampled natural seedlings and selected seedlings from both zones in approximately equal numbers. And we used 4 primary data sources from approximately 1,000 seedlings of natural and 1,000 seedlings from selected sources. We used genotypic data from 50,000 SNP array. We used phenotypic data for 4 traits: height growth, autumn cold injury, growth initiation, analogous to bud break, and growth cessation, which is analogous to bud set. We also integrated this with 19 climatic and 3 geographic variables. And we also have a mapping population that we used for some of the latter analyses from a single outcross from an 106 haploid F1 progeny. The objective is to be able to make comparative analyses of positive effect allele frequencies and linkage disequilibrium between natural and selectively bred seedlings. The processing steps are outlined here. If you would like more detail on them, come and see me later. But the core of our analyses is this genotype-phenotype association, also known as GWAS, and we did this performed this in the natural seedlings only to avoid biases from population genetic structure and to understand how selective breeding is affecting naturally occurring variation, naturally adaptive variation. We also used phenotypic data in terms of breeding zone means. Climatic data we converted to breeding zone means. And we also used mean pairwise correlations among contigs from the mapping population. So positive effect alleles. Quite simply, from the genotype-phenotype association, it is the positive effect allele is the allele that has the effect of increasing the expression of a trait. In this case, positive effect alleles are greater height, increased autumn cold injury, delayed growth initiation, and delayed growth cessation. And our use of this approach is based on some of the work for human height. Now, these studies on human height in Europe had some fairly substantial shortcomings. We've avoided those by using just the GWAS of the natural seedlings, but we're using the general approach of alleles that have a positive effect on the phenotype and looking at the shifts in their distributions. Okay, so in the process of doing this analysis, we made some regional comparisons as well that I'm going to show you for the positive effect allele analyses. We identified and separated out the results for breeding zones in southern BC and breeding zones in central BC. From previous work on the phenotypic data from these populations, we found there were probably substantial differences between these two regions. So I'm going to focus on the results for height and cold injury first. And this is a histogram of, on the x-axis, we've got the frequency of SNPs, frequency of positive effect alleles, I should say, associated with height. And on the y-axis, we've got the frequency of seedlings with a given number of positive effect alleles. And so this is simply calculated as the number of positive effect alleles in each seedling across the top 186, the top 1% of SNPs from the genotype-phenotype associations. And what we see here for height-associated SNPs in both southern British Columbia and central British Columbia is a systematic shift in the number of positive effect alleles that seedlings have towards an increased number of positive effect alleles for height growth, indicated by these blue versus red curves. For cold injury associated SNPs, we see the same general trend. We see that selectively bred seedlings on average have an increased number of positive effect alleles associated with increased cold injury. These numbers are consistent but generally not large, but provide detectable results. On average, there are about 8 to 12 additional positive effect alleles in natural versus selected seedlings for both these traits. Now, when we flip this around and look at the number of positive effect alleles per SNP across individuals, we see these results. On the x-axis, we have the top 1% of SNPs from the genotype-phenotype associations ranked from the most significant to the least significant. And on the y-axis, we have the difference in positive effect allele frequency for a given SNP, calculated as the frequency of positive effect alleles in the selected seedlings minus the same value for the natural seedlings. And so what we see is several patterns. Firstly, the SNPs that rank as most significant in the genotype-phenotype associations consistently have a strong positive response to selective breeding. And this is the case in both traits and in both breeding zones for height, cold injury, British Columbia South, British Columbia Central. Now what the red bars here indicate are SNPs that overlap in the top 1% by, of all 4 traits. So they overlap by identity. And you can see that they constitute a large proportion of the response to selection, and they dominate the SNPs with the strongest genotype-phenotype association in all traits. However, the remaining 100 SNPs in the 186 have variable responses to selection, and some of these can be quite strong. The implications for this are that even though the average number of SNPs per seedling only increases by 8 to 12, all SNPs within the top 186 are capable of responding to selection, suggesting that there is a substantial amount of redundancy among SNPs within those that are significantly associated with a trait. So now, looking at the range-wide analyses again, this is a plot of linkage disequilibrium, and below the diagonal we've employed the mapping population, and for the SNPs in the top 186 that we could map to the loblolly pine reference genome, we have calculated the pairwise correlation, squared correlation between contigs, and clustered those pairwise correlations. And what this does, because it's from the mapping population of control cross, it gives us an indication of what the structural association of these contigs and the loci we're interested in, what their associations are within the genome. And we can see that there's several clusters of correlated pairwise correlations indicating that the SNPs of interest are distributed throughout the genome. And we see this for both height-associated SNPs and cold injury-associated SNPs. However, there are also some outlying clusters when we arrange the natural pairwise linkage disequilibrium values on the same order as the mapping population, and I'll come back to the reasons for that in a minute. So to summarize the height and cold injury data, there are systematic shifts in the frequencies of positive effect alleles associated with selective breeding for increased height, and we see this for both height and cold injury. There appear to be multiple variants that can contribute to this, and multiple SNP variants that can contribute to this. And while they're concentrated in the SNPs that are most strongly associated with the genotype, phenotype associations, all SNPs that we surveyed in the top 186 are capable of responding. So it indicates that responses to selection are not limited to those SNPs most strongly associated with the phenotype. And therefore, we see some evidence that genomic redundancy is apparent within these results. And the genotype-phenotype associations identify SNPs throughout the genome as indicated by the linkage groups we've found. So now to look at the results for growth initiation and growth cessation, we see these very distinct multimodal patterns in terms of positive effect allele frequencies within seedlings. We see this for both British Columbia South, British Columbia Central, and for growth initiation SNPs and growth cessation SNPs. What I should point out here is that the difference between natural and selected for British Columbia South is actually quite a lot greater than it is for British Columbia Central in terms of selected versus natural seedlings. There's approximately 20 to 25 more positive effect alleles in British Columbia and selected seedlings from British Columbia South than there are from British Columbia Central. The reason for this is in this plot here. When we look at the responses of individual SNPs, we see there is this wholesale change, wholesale increase in the frequency of SNPs from British Columbia South that does not occur in central British Columbia, suggesting that a large number of SNPs are responding to selection that does not occur in the central region. And again, when we look at the patterns of linkage disequilibrium in the natural populations versus pairwise correlations among contigs in the mapping population, we see that there is a strong structural basis and a strongly concentrated genomic architecture for the patterns we see in terms of responses to selection. But there are also outlying clusters here, suggesting that selection is also generating patterns of linkage disequilibrium independently from structural associations within the genome. So to summarize, there are multimodal frequency distributions of positive effect alleles, but only mostly in BC South are the systematic shifts in the frequency of these alleles. The wholesale increase in SNP frequencies appears to underlie a strong response to selection for height, and physical linkage and selection drive the patterns of drive genomic architecture which underlie the patterns or the responses to selection we see. However, between seedling types, I haven't shown the results here. The results for linkage disequilibrium were identical. There was no significant change that we could detect between natural and selected seedlings in pairwise linkage disequilibrium. So to look at this, summarize this in one final way, looking at multi-locus clines. All we did here for each breeding zone and each seedling type was calculate the total number of positive effect alleles across seedlings and across SNPs. And to summarize on the y-axis, the positive effect allele frequency, while on the x-axis we just used the mean climatic variable for each breeding zone. And what we can see is that these genomic clines are really very strong in all cases, but there's very little response to selection in any of them. The strongest association is with extreme minimum temperature, indicating that this is the most important trait for defining local adaptation in this species. And we've seen this repeatedly in other studies. Likewise, for phenology traits, we see the same patterns. Extreme minimum temperature has the strongest multi-locus genomic associations. So the conclusions and implications from this are multi-locus signature selection are detectable in lodgepole pine even after just 2 generations of selection. And the genomic architecture of the traits we're interested in varies substantially. But at present, it appears from the clinal analyses that the trade-offs among these traits are maintained sufficiently to maintain local adaptation, despite the fact that the genomic architectures indicate that there is potential for trade-offs, as we see in the differences between British Columbia South and British Columbia Central in responses to selection. In relation to assisted gene flow, the really strong genomic clines we see suggest that assisted gene flow is warranted to manage the adaptation of seedlots to future climates. But we don't really see any difference in the significant differences in the clines on a range-wide level, which suggests that natural and selected assisted gene flow prescriptions can probably be applied in the same way. We don't need to differentiate between the 2 seed sources. And lastly, positive effect alleles seem like a really simple, sensitive, and effective basis to rapidly assess local adaptation to climate, because they just act as a good summary statistic. And that's something I'm looking at taking forward in the future, and maybe refining how to identify those positive effect alleles, and maybe whether we can use effect sizes as well instead of p-values to determine which SNPs to use. Okay, so this project was going on a long time. There are a lot of collaborators that I've been working with and would like to thank. Any questions?
We have time perhaps for 1 or 2 questions. Yes, Santi?
Yeah, I was wondering if for you have considered to include also effect size in when you do your count of beneficial alleles. It would not be, I understand you gave the same weight to all kind of, or then assuming that they have similar effect size, but if you are doing GWAS, you will not, that some are having more impact than others, so.
So I did run these analyses weighted by effect size, and it didn't really make any tangible difference, although I have looked at the effect size data and I have plans to use that going forward in the future. I'd like to talk with you more about it if you want to catch up with me afterwards.